Hi, this is Dr. McShafery, and I wanted to create a short tutorial for the leaf structure lab, part of the leaf and pigment lab. I know sometimes during lab people have trouble seeing some of the structures under the microscope, so I wanted you to be able to use this as a review. So let's start with the basics of leaf structure. On the outside we have the cuticle. It's waxy and it waterproofs the leaf. Underneath that we have the epidermis. Now the epidermis is just a single layer of cells. It's on the leaf surface and it serves to protect the leaf. It's also what secretes the cuticle. Now there are some structures in the epidermis, specifically the stomata, which are openings in the epidermis that will let CO2 in and let oxygen out during photosynthesis. And these stomata are protected by guard cells. And these are a pair of epidermal cells that are specialized. They can close whenever the plant is low on water, and that will keep the plant from wilting. If there's plenty of water, they'll open up and let CO2 in, but of course at that time they're also letting water out. And then we have all the stuff that's in the middle. The mesophyll literally means the middle leaf. And it's everything in between. It's the Oreo and the cookie, or the white stuff in the Oreo cookie. So we have two types of cells where most of the photosynthesis occurs. First are the palisade mesophyll cells. These are stacked real close together, and the vast majority of photosynthesis occurs right there. But there's also spongy mesophyll, and that is kind of loosely packed, and that helps the stomata get air to the palisade mesophyll, and you'll see that in a minute. We also have vascular tissue in there, and that either is xylem, which carries water and minerals to the leaf, or something that carries sugars to and from the leaf, and that would be the phloem. Then there are fibers, which are very thick-walled cells that strengthen the leaf, and there are a couple other types of cells that are in the middle of the leaf, sometimes for storage of water or sugars or something like that. So in lab we have a model, and you might want to take a good look at that when you're in the lab. And you can see here, the leaf is, again, like a sandwich. Now, there may be more layers of palisade mesophyll or spongy mesophyll, but basically it looks like this. You have cuticle right here on the outside. Then you have epidermis. These are these cells right here, and those lie under the cuticle. Then we have the palisade mesophyll. That's all these cells here that are stacked closely together. And then we have what is called the vascular bundle. Okay, and the vascular bundle is where the xylem and the phloem are at. Okay, there are bundle sheath cells around the outer edge of it, and then inside are packed the xylem. Okay, that would be these cells that are in red here. That's what carries water and minerals out to the leaf. And then we also have phloem. Okay, and the phloem lies here, and these cells are actually separated with little perforations. There's a lot of other interesting differences between xylem and phloem, but we won't go into those now. And then, of course, the spongy mesophyll is on the other side of the leaf, kind of from the palisade mesophyll. You can't see behind this, but behind the vascular bundle there, the spongy mesophyll would run into the palisade mesophyll. Okay. Now, one common problem in lab is that the slides aren't always the same. And sometimes, to complicate matters, you might put them in upside down. And so the first thing you need to do is get yourself oriented. Usually, for most plants, the palisade mesophyll is going to be on the sunny side, the top side. Okay? The spongy mesophyll and the stomata are usually more common on the shady side. Stomata can often be found on both sides of a leaf, but they'll generally be a little bit more on the shady side where they're not going to be as subject to evaporation. If you have a floating aquatic plant, the stomata are primarily going to be on the upper side, and submerged aquatic plants may not have any stomata at all. And not all plants have both spongy and palisade mesophyll. So let's take a look at two examples here. This is the water lily here on the left. It has large air spaces in there. You can see the vascular bundle here, which we'll talk about in a minute. And then up here you can see what passes for both spongy mesophyll and palisade mesophyll at once. And it's up there that the stomata are going to be opening up into these air spaces, because that's the part that's facing the air. The bottom side is facing the water. If we go over to the syringia, also called the lilac, you can see all the palisade mesophyll up here. Here's the epidermal layer. And then the stomata are going to be places like right there, okay, where the air can come in through what are called guard cells. And we'll also talk about those later. And then you have a big vascular bundle here. That's the midrib of the leaf. Okay. In lab, we talk about three types of plants. We have aquatic plants, also called hydrophytic. They may be completely submerged, they may float on the surface, or they can be what we call emergent. Their roots are down on the bottom of the body of water, and part of the plant sticks up above. If they are submerged, they usually don't have a cuticle or stomata. They just don't need them in the water. If they are floating, the stomata and the palisade mesophyll will generally be on top, and again, they may also have air pockets to help them stay on the surface. Okay, mesophytic. Meso again means middle, and so these are plants in the middle. They live in environments with sufficient water, but they're not completely submerged or actually in the water like a hydrophytic plant would be. And these generally will have a thin cuticle. They will have a palisade mesophyll that is on the side exposed to the sun, and most of the stomata will be on the opposite side. Now, some mesophytic plants like grass, which grow straight up, will have a fair number of stomata on both sides, because it depends on which way the leaf is leaning as to what's the top or the bottom. And then we have the xerophytic plants. Okay, these are the ones that live in very dry environments, and they're going to have a thick cuticle. The slides we have in lab are stained. The cuticle is usually stained red, and you can see that. They often don't have distinct palisade or spongy mesophyll. These cells that are doing the photosynthesis are still mesophyll cells. They may be on both sides of the leaf. They may just be on the top side, but they will be present there, but usually not organized like we see in the mesophytic plants. The mesophyll may have a lot of water storage cells in there, because that's going to be important. And since that leaf is very thick and heavy, the vascular tissue is often reinforced with thick-walled fiber cells. And overall, you're probably going to expect to see fewer stomata. They're probably going to be away from the sun, and sometimes they're in sunken pits. And again, all these are rules that can be broken, and there's plants that fall in between all these categories. So now I'm going to take you through some of the slides. These are photographs of some of the slides we use in lab, and these are cross sections. They took a leaf and they took a very thin slice out of it, going from one side of the leaf to the other, and that's what you're looking at under the microscope. This is Elodea, which is an aquatic plant. There is a very thin cuticle, if any cuticle at all, on the outside. You don't see organization into like spongy mesophyll or palisade mesophyll. Most of the cells have chloroplasts in them, and the one they're showing here only has 2 rows of cells. Some of the live cells we look in class have more layers of cells. And you can see the vascular bundle right here. It's not very well developed because, again, you don't need a lot of xylem in a plant like this because all of the cells can get water. Okay, a little bit closer look at that center part. And again, we're not seeing any stomata. We're just seeing those vascular bundle cells right here in the middle, and then these larger cells around the outside. Okay, one more view. I believe this is the top side from what I can tell, and then these smaller cells are on the bottom side. These cells are filled with water, they're not filled with air. This is not a floating plant, but it is an aquatic plant. Okay, now we're going to switch. This is the water lily, Nymphaea. And this one is a little bit more organized. Okay, we have a good vascular bundle here, and again, that's the midrib of the leaf. That's the very thick vein in the middle of the leaf. And up on top, these big cells here, that is the xylem. And this down here would be the phloem. Okay, the phloem again is moving sap around, the xylem is moving water. Again, the xylem doesn't have to be really well developed here. It does have kind of thick walls to help strengthen the leaf. Okay, these big open spaces, these have air in them. Okay, that helps the leaf stay at the surface. And most of the action here is going on up at the surface. Okay, these would be the cells that are photosynthetic, particularly the ones where all that dark stain color there, those are the really photosynthetic cells. And if there are going to be stomata, they're going to be up there. You've got more air spaces here, and so many of these will have guard cells at them, and that's where the stomata will be located. But again, that's the top up there, so this thing is right side up. Okay. Another look at it, just a little bit closer look at the thick-walled. You should recognize it now, the xylem cells, and then all these in here are the phloem cells. As the plant produces sugars, those will be moved through the phloem down to the roots. Okay, and yet another look, and we want to zoom in a little bit at the top, and here and here, these are guard cells. This is our first close look at them. And again, this is on the upper surface of the water lily leaf. And they open up this hole in between them. Bring that up a little bit. That's the stomata. Or in this case, since there's just one, that's a stoma. Okay, here is another one here. Okay, that's the stoma, the hole. The plural stomata. And then the cells to either side, here and here, those are the guard cells again. When the plant is happy, those cells get small and open up the stoma so that air can move through there. When the plant is sad, like when it's out of water, they swell up, as you see here, and they close that off. Okay? They're not always all open at the same time, and I think these are actually just sliced at a slightly different point so you don't see the opening there. These are actually chloroplasts here that I think got sliced out when they ran the razor through this. Okay, we're going to switch now to the xerophyte, and this is the yucca plant from the desert Southwest, although we have them growing in Ohio as well. Very different pattern. Okay, you'll notice that it has a very thick cuticle out here on the outside, and the vascular tissue is scattered in these vascular bundles. And again, you are going to have xylem and phloem in there. With these bigger cells would be the xylem, and these other cells are going to be the phloem. And then you are going to have these cells up above. These are going to be the fibers. And those make the leaf very thick. And you can see as you get to the outside, you'll get to the point where there's not even any vascular tissue there at all. It's just fibers to make the leaf strong. And all these cells in between are storing water. Okay, close-up look at the vascular bundles, xylem down here, phloem up above, and the fibers. Very thick-walled. They don't really have much going on inside them when they get this developed. Okay, on the on the yucca plant, the stomata are found, and they are actually down in little pits. Okay, so they're down in there. There's the stoma, and then on either side of it are the two guard cells. And they are down in little pits in there, so that, and they just open up into a small area. There's not a lot of air space in there. These plants photosynthesize very slowly, and again, most of the photosynthesis goes on near the surface. So the carbon dioxide doesn't need to get deep into the plant, and that cuts down on evaporation a bit. Okay, here's another view, and you could see the two guard cells, and this one is closed. And again, the air could come in through here. By putting it down in a little pit, that protects it from the wind a little bit, and that cuts down on evaporation even more. And again, you could see very thick cells up there that are making up the fiber bundles. Now finally, we're going to talk about the lilac, the syringia. And this is the typical mesophytic plant. So we've looked at the extremes. We looked at the two aquatic ones, the hydrophytic. We looked at the xerophytic yucca. And now we have the lilac, which is the one in between. And so this is kind of a big view of it. And you could see the middle part here, the vascular bundle. You could see here good clear palisade mesophyll. Down here, this is spongy mesophyll. You've got epidermal cells on the top and on the bottom. So that's kind of the overview. You can see the air spaces in between the palisade mesophyll. There are some air spaces up there. There are a few stomata up there, and then there are a number on the bottom part of the leaf. Okay, zoom in a little bit. This is a small vascular bundle off to the side. It's not the main one. And then this is a branch over here that's coming off it and running out to the side of the leaf. And you can just see little bits and pieces of it. And again, up top, you've got the palisade mesophyll, in this case 2 layers of it, maybe 3 in some places. You have the epidermal cells up there, a little bit of cuticle, not too much. And then down at the bottom, you've got the spongy mesophyll, and in places that you've got stomata going up in there. And we'll go a little bit further here. And this is a close-up, and you can see real clearly here the stomata and the 2 guard cells. And that central opening, that was the stomata. And then you have this, all this in here is airspace, so CO2 can get through here and work its way up to those palisade mesophyll cells, which are very reddish, and that indicates, with the stains they used here, that there's a lot of chlorophyll. Okay, that's it for this part. I'm going to then put some videos on of cytoplasmic streaming in Elodea, which is something we always look for in lab, but we don't always see. Okay, this portion here is going to show Elodea, and you're going to see cytoplasmic streaming. And as you can see in the picture here, those little green things sliding around, those are the chloroplasts. And what's happening is inside the cell, there is a cytoskeleton. And it's made out of actin and myosin, just like your muscles. And the chloroplasts are attached to those, and they shorten and lengthen and drag the chloroplasts around the outside of the cell, outside of that vacuole that's in the middle. And so you're seeing those move, and what the cell is trying to do is make sure all the chloroplasts get into the light, that they're able to get CO2 and whatever else they need. So we'll just watch a little bit of this, and you'll see it moving around. After a while, it picks up a little bit, and certain parts of the plant go a little bit faster. Depends on how much light they're getting.